degrees. Next minute it's 35 degrees. I was scraping ice off of the car this morning. Crazy, crazy, crazy. All right. We are going to do the benchmark test next week. I do want to finish this unit. Let me pull this up first. Still waiting on somebody to come to class, though. All right, let me. I'll bet. I'll bet. Yeah, and I had gotten to the point I was opening the window in the room at night. I was glad I closed it last night because I would have been freezing. But yeah, this, I don't understand. And in my environmental classes, we're talking about global warming. This is global warming for sure, for sure, for sure. All righty. Okay. So, grades have been posted. So the grade you have that you see in Canvas, today's the last day, you're probably gonna be able to see that anyway, because they change over. As of today, grades will be, everything else will be on the next quarter. I'm looking at Canvas, so got two things going on in my head at the same time. The mouse lab that you are working on in Gizmo, that's going to be the first grade in the next semester, next quarter. So that grade will not show up. I didn't want to have to deal with zeros and give people a chance. So Monday for you all, Tuesday, I guess, is the last day that that will be late on Tuesday, and we will move on. So what I want to look at today, we talked about genetic disorders a little bit when we were talking about chromosomes earlier. So we will kind of look at this very quickly, and then we're going to watch the Amoeba sisters talk about karyotyping. And these are karyotypes. And I just wanted to make sure I went over this with you because it might be on the benchmark. It will definitely be on the milestone, but not a lot. You just need to be familiar with what a karyotype is, be able to read one. So we'll deal with that in a little while. All right, so let's take a look at this.
An overview of some common genetic disorders. Dominant allele disorders. Achondroplasia is a dominant condition in which the long bones do not develop properly, resulting in unusually short arms and legs, a large head, and a relatively long torso. Most cases of achondroplasia occur spontaneously as the result of a new mutation in an egg or sperm of one of the normal appearing parents. Only 1/8 of the children born with this condition inherited it from a parent. Huntington's disease is a progressive degenerative disease that can cause certain nerve cells in your brain to waste away. As a result, you may experience uncontrolled movements, emotional disturbances, and mental deterioration. Huntington's disease is inherited as a dominant allele. If one parent has this disease, the offspring have a 50% chance of inheriting it. Recessive allele disorders. Cystic fibrosis is an inherited recessive disorder resulting from a mutation of the gene that regulates the secretory gland, including mucus and sweat glands. The characteristics of cystic fibrosis are salty tasting skin, normal appetite but poor growth and poor weight gain, excessive mucus production, frequent chest infections, and coughing, shortness of breath. Both parents must carry the allele for it.
All of a sudden, she stopped reading. You might have heard of Tay-Sachs disease.
Albinism is a recessive disorder that occurs when one of the several genetic defects makes the body unable to produce or distribute melanin, a natural substance that gives color to your hair, skin, and iris of the eye. In one type of albinism, people have white or pink hair, skin, and iris color, as well as vision problems. Another type of albinism, called ocular albinism type, affects only the eyes. Sex-linked disorders. Hemophilia is a disorder that impairs the body's ability to control blood clotting, which is used to stop bleeding. Because it is X-linked, only males typically exhibit symptoms. Muscular dystrophy refers to a group of X-linked inherited diseases marked by progressive weakness and degeneration of the skeletal or voluntary muscles that control movement. Duchenne muscular dystrophy is the most common and severe childhood form of the disease. Color blindness is an X-linked condition that results in the inability to perceive differences between some of the colors that others can distinguish. There are several types that enable some individuals to perceive different colors than other affected individuals. Some cannot distinguish between red and green, while others have the inability to distinguish between blue and yellow. Chromosomal disorders. Klinefelter syndrome is a condition that occurs in men who have an extra X chromosome in most of their cells, XXY. The syndrome can affect different stages of physical, language, and social development. The most common symptom is infertility. Because they often don't make as much of the male hormone testosterone as other boys, teenagers with Klinefelter syndrome may have less facial and body hair. Turner syndrome, a rare disease that only affects females. It is caused by the abnormality in one of the girl's X chromosomes, leaving her with only one fully functional X chromosome. As a result, girls with Turner syndrome tend to be sexually undeveloped and infertile. Other common symptoms of Turner syndrome include a short stature and a webbed neck. Down syndrome. Children with Down syndrome have some of the following physical traits: short stature, weak muscles, and a short, wide neck with excess fat and skin. Small, low-set ears, an irregularly shaped mouth and tongue, and mild to moderate mental disability or retardation are also characteristics of Down syndrome. An extra 21st chromosome is responsible for this disorder.
Okay, so those are really only the most common ones that you'll probably hear. There's a lot more chromosomal diseases. So again, this karyotype is a map of all. If we took all the chromosomes in your body and laid them out, you have humans. Every species has a different number of chromosomes. We know you're a human because there will be 46 chromosomes, and they number them. And 46 individuals, but 23 pairs. And so again, when we talk about alleles, you have two alleles, one from each parent, and they match up. So they line up together. So like number one here, one is from mom, one is from dad, but they have the same information on them. And then we come down to this one down here. So they put this where you could see if you are a male, you have an X and a Y, or if you're a female, you have X and X. Okay, and that's where all of the characteristics for femaleness or maleness will be located. All the rest of these are what we call autosomal chromosomes. They have the characteristics for everything else in your body: your eyes, your ears, your height, your the fingers and toes. All of that is in one of these other 22 sets, and then maleness and femaleness is on the 23rd set. Okay. So this is what they're saying a normal would look like. Someone with trisomy, this one right here, on the 21st, there's 3 of them, and that's not what it should be. Okay, so trisomy leads to Down syndrome. And I think when we did genetics, when we did DNA, we looked at some that had one was missing, or there was a defect in one of the chromosomes. So we have this list here, and I'll get back to that in a minute. What I am going to do is let her talk about talk.
Talking about biology, gotta be careful about diagrams. For example, view a circulatory diagram and you might think human blood can be blue or red, not realizing that human blood is red and the color coding used is showing oxygen concentration instead. Or see a diagram of a plant cell and suddenly you think all plant cells are boxy and all of them are not. Enter the chromosome. Diagrams for the
Buffering. But yes, your blood is always red. It's either dark red or bright red, depending on if you have a lot of oxygen in it, then it's bright red. If it's what we call deoxygenated, it'll be dark red. And I don't know why it's spinning round and round. There we go.
chromosome have done me wrong too. For example, I remember often seeing chromosomes drawn in an X shape. So I thought, okay, all chromosomes are in an X shape, which was really confusing when I heard about people having an X and a Y chromosome, because then I started to wonder, why are these chromosomes resembling the letters of the alphabet? And then sometimes they look like single stick pieces and not X shapes, and I wondered, what is going on with these things? I also thought that if in most cases humans have 46 chromosomes, then they're in there somewhere deep inside the body, that if you need to analyze them, you'd have to find them and make sure not to do anything to them because that's all you got. Didn't realize you find those chromosomes in nearly all body cells. So those were some misconceptions I had. We're hoping to clear those up and more about chromosomes. In this video, we're going to focus on eukaryotic chromosomes. And specifically, we're going to focus on human chromosomes. Chromosomes are made of chromatin, and chromatin
All right, I'm
is
going to
made...
pause it there. Because we are reviewing for benchmark, all those kinds, all these words of vocabulary words, you need to be securing them in your head. Eukaryotic cells mean those are the cells that have a nucleus. Okay. Prokaryotic cells are the ones like bacteria that don't, bacteria and viruses that don't have a nucleus. They have DNA, but they're not connected in a nucleus. All right. So be listening to this for vocabulary for review as well as the new information about karyotypes.
A chromosome itself is really intense packing of chromatin. It starts with DNA being wound around proteins called histones. This forms nucleosomes, which are often considered to look like beads. More and more condensing packaging levels occur until it results in the chromosome level. But why all this packing? Well, in your cells, DNA has to fit inside a nucleus. It's also really important for when you make more cells, like in the process of mitosis. In mitosis, you have to be able to move DNA into new daughter cells. You have so much DNA that these highly packed chromosomal units make it easier to do so. It's important to remember that within the same human body, the chromosomes in one body cell are generally the same in another body cell. So, for example, if you were to compare a stomach cell and an eye cell in the same body, the chromosomes in those two cells are generally the same. Certain genes on the chromosomes may be turned on or off by a variety of factors. You wouldn't want your eye cells actively using the gene to produce stomach acid. That would be not ideal. By the way, DNA is not always coiled up tight. For example, in DNA replication, DNA typically needs to be unwound. There are a variety of factors that influence how tightly packed the chromatin is. Let's get kind of a general idea about how human chromosomes may be represented. Human chromosomes can be like this as a single component, or they can be replicated like this. This one has two identical chromatids called sister chromatids. When these two sister chromatids are attached, it's still considered one chromosome, but it has two copies. When these sister chromatids are separated, each sister chromatid can be called a chromosome. So this is a chromosome, this is a chromosome. In most cases in humans, each body cell has 46 chromosomes. In interphase, the DNA is copied, which will result in duplicated chromosomes. There's still 46 chromosomes here, but that's 92 chromatids in this picture. During a stage of mitosis, these 92 chromatids get pulled apart, so that each cell will have 46 chromosomes. We have an updated chromosome numbers video to help clear that up a little more. And in our updated chromosome numbers video, we also mention more about the centromere, and we point out that you can recognize the term centromere as a traditional physical definition of a constricted region, as you've likely seen in diagrams that look like this, a region where the sister chromatids would be attached. But also, a centromere can be defined as a specific area of DNA and the proteins that bind there. Each of these sister chromatids technically have a centromere, which makes sense. I mean, these sister chromatids are identical copies. So you may also see diagrams that try to emphasize that. By the way, the centromere region, despite its name sounding like center, does not have to be in the center of a chromosome. So maybe you've seen pictures of all those human chromosomes before in a caption that reads human karyotype. Karyotypes represent the appearance of an individual's chromosomes, the number of them, size, where the centromeres are. Cytogeneticists are examples of scientists that do karyotyping. It often includes placing isolated body cells, which could be from blood or body tissue, into a certain hypotonic solution. If you recall from our osmosis video about hypotonic and hypertonic, if the solution is hypotonic to the cells, that means the cells are hypertonic. Water is going to have a net movement into them, meaning the cells are going to swell with water. And that will help the chromosomes spread apart for karyotyping. The chromosomes can be stained and all the chromosomes can be identified and arranged. You can produce an image from this. To learn more about karyotyping and producing the images, please see our video description. The image itself produced can be called a karyogram. Just to note, it's common for many sources to use the term karyotype as a general term for both the description of the chromosomes and the visual image that may be produced and included. However, more technical sources tend to distinguish the visual picture as a karyogram, which tends to be produced as part of karyotyping. If a scientist is going to do karyotyping, which often will include a produced image along with it, the chromosomes need to be visible. Visibility is good. And that impacts when karyotyping is done. If you tried to take a karyotype during interphase, for example, that would be a challenge because you don't have that condensed DNA packing, that ideal chromosome visibility. For optimal viewing, a typical karyotype is taken at or right before metaphase. Recall from mitosis that in metaphase, each chromosome would have 2 joined sister chromatids. So if you search for karyotypes online and you come across differing images, maybe like these, remember, a typical karyogram does have 2 sister chromatids per chromosome. It's just that many times the joined sister chromatids of each chromosome can be so close together that to an untrained eye, it can be difficult to tell. In the process of karyotyping, chromosomes can be arranged in homologous pairs. Homologous chromosomes are about the same size and contain the same types of genes, and within each homologous pair, you receive 1 chromosome from one biological parent and 1 chromosome from the other biological parent. In this general example of a typical karyotype of 46 chromosomes, you could see 23 pairs. 22 of the pairs, which would be 44 of the chromosomes, are called autosomes. This means these chromosomes are not related to biological sex. They may have genes related to eye color or height or hair texture. The last 2 chromosomes are called sex chromosomes because they are related to biological sex. Sex chromosomes are the chromosomes that get named X or Y. The name X and the name Y that are used for sex chromosomes have nothing to do with the shape of the chromosomes. The reason is an interesting topic to look up, though. In most cases, females have 2 X chromosomes, and in most cases, males have 1 X and 1 Y chromosome. But as we mentioned in our Punnett squares and sex-linked traits video, it is possible to have fewer or more sex chromosomes. More about that in our description. So in this example karyotype, 23 of these chromosomes are from a sperm cell from the male biological parent, a sperm cell which is a gamete. Unlike body cells, gametes have half the number of chromosomes as body cells. Sperm cells are considered haploid because they only have 1 set of chromosomes. The other 23 of the chromosomes came from an egg from the female biological parent. An egg is also a gamete. Egg cells are haploid because, like the sperm cell, an egg cell contains only 1 set of chromosomes. When a sperm cell and an egg cell combine, they form a fertilized egg, known as a zygote. The zygote is diploid because it has 2 sets of chromosomes, and it will divide to continue to form more diploid cells. When it comes to sex chromosomes, generally females can only provide an X chromosome, as that's the type of sex chromosome they can give. And generally, males would have the ability to give an X chromosome, generally resulting in a female baby, or a Y chromosome, generally resulting in a male baby. Because sperm cells can carry an X or a Y chromosome, this is why males are considered to determine the biological sex of the offspring. Interesting fact to add about the Y chromosome has a gene on it called the SRY gene, which stands for Sex Determining Region Y. And it is a gene that, when expressed, triggers the development of male biological characteristics. So why do karyotyping? Why have detail about a person's chromosomes and also have it visually shown? Well, it could be for medical screening, for example. There are conditions where an individual may have more or less chromosomes. For example, 47 chromosomes or 45 chromosomes, or individual chromosomes themselves could have missing or extra segments. Karyotyping can reveal this. Do you have an interest in a career that involves this area of study? The field of genetics continues to expand, and the career of a genetic counselor may be something to read up on. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, back to. All right, so there were a lot of vocabulary words in there. I wrote down 10. Eukaryote, we talked about it at the beginning. Those are these type of cells that have a nucleus in the center, and those are what we have. Homologous pairs, meaning they are the same. We have that homozygous and this, it means the same. So homologous pairs of chromosomes mean that when you have the lineup of chromosomes, the information on each one is the same. So you look at these, the bands on each of these chromosomes line up. So the chromosome that you get from mom and the chromosome that you get from dad will have the same information and they will line up. Okay. So they contain the same information. That's homologous pairs. The term alleles, and again, I'm reviewing all these terms. Each one of these is also called an allele. Okay, an allele is a chromosome with specific characteristics. Okay, so these are all alleles, but each one of them is different. So chromosomes is a general term. They're all chromosomes, but each one is an allele. That's a way that you can distinguish those two words. Autosomes, I wrote down, those are the ones again in your body. So all 22 of these would be autosomal chromosomes. These last ones are the sex chromosomes. Gamete, the sex cells in your body. So the cell that will determine male or female is an egg cell for females, a sperm cell for males. Hopefully you knew that. Those types of cells are haploid, meaning they only have half the number of chromosomes. So if you took this picture and again, you went down the middle during meiosis, these would separate and one will go to one place and one will go to the other. And each gamete will have half the number of chromosomes so that when the egg unites with the sperm, mom's 23 unite with dad's 23 and we're back to 46 chromosomes again. Okay. Zygote is a fertilized egg and it again will have the diploid number, meaning full set. It sounds like two, but diploid in this case means a full set of chromosomes. So haploid is half the number, which would be 23. For us, diploid is 46. All right. I want you to, if you've completed your mouse gizmo, I want you to go ahead and look at these. Again, we've talked about most of those. And I have not assigned this. Let me see if I want to. I tell you what, instead of doing, let me go ahead and publish this one. All right, we're going to publish it. I'm going to make it's available. It's available today. It's going to be due next when, actually no, because we're going to do benchmark. So, yeah, I can make it. It'll be due next Friday. And I'm leaving it open till the end of the month. All right. So it's already in your module. I just had not published it. So I didn't know whether we were going to get to it or not. Come on, save. My computer is frozen. Oh, I didn't like, what's wrong with this? How did I get to that date? Okay. All right, so you should find this in your module now. Katie, you're still here. Katie, you should be in your fourth period class. You're being marked absent in your fourth period class. All right, she's gone. Okay, so download this worksheet and it looks just like the one we were showing you. And all you have to do is to be able to read through. So they have numbers underneath each one. And you're going to decide, and I just went through all the vocabulary. So you know what homologous pairs are, you know which one, you know what autosomes are, and sex linked. Okay, so you're going to label those and then you're going to use this box over here to answer these questions over here. Okay. Actually, hold on. These questions go with that up there. This right here, this is figure A. This is figure B, C, D, and E. So these questions want you to look at, so you're gonna have to look real close at each of the chromosomes and figure out what you see. They're all labeled for you. Figure out what you see from what you don't see based on the fact of the information that you've had up here. You're going to decide not only are you going to decide whether it's male or female. So you're naming the disorder that the person, whatever's going on over here after you've looked at what's happening up here. And then you're going to say whether the individual in the picture is male or female based on whether or not they have X and Y chromosomes. Okay. You're always going to be looking to the last ones to decide whether they are male or female. All right. Okay. Finish the mouse gizmo first. Everybody should be finished with that and then work on this one. So you might have to refresh your page. It was there, it just wasn't published. Okay. All right, I'm going to stop sharing so I can do attendance. And how's it going, everybody? Any questions? Any questions? Yes. All right, lovely people. It is time. You all have a great weekend.